This protocol displays the use of a novel microfluidic co-culture model that enables independent culture of eukaryotic cells and bacteria to test the effect of the commensal microenvironment on pathogen colonization. The co-culture model is demonstrated by developing a commensal E. coli biofilm among HeLa cells, followed by introduction of enterohemorrhagic E. coli, or EHEC, into the commensal island in a sequence that mimics the sequence of events in GI tract infection. The resulting extent of epithelial cell colonization by EHEC is visualized using fluorescence microscopy.
Hi, I'm Jeongyun from the Laboratory of Molecular System Biotechnology in the Department of Chemical Engineering at Texas A&M University. Today, we will show you a procedure for cultivating bacteria and eukaryotic cells together in a microfluidic device, in a manner that mimics their organization in the human gastrointestinal tract. We use this procedure in our laboratory to study how pathogenic bacterial colonization of the epithelial cell is affected by signals that they encounter in the GI tract prior to colonization. So let's get started.
Prior to experimentation, prepare a silicon master, designed and fabricated according to intended experimental use. For example, the Stanford Microfluidics Foundry will fabricate designed masters from which subsequent experimental replica molds can be made. Molds for 2 membranes, for the eukaryotic cell culture layer and the pneumatic layer, and 1 bacterial channel are required for the tri-layer device described in this protocol. The channel heights for the bacterial, pneumatic, and eukaryotic cell culture layers